Okay, so we're still on the cell membrane or the plasma membrane, and we're talking about different ways to cross over the membrane. And if we look at the top of the image there, simple diffusion is just going right through the membrane, right? So oxygen and CO2 do that. So it's passive, requires no energy at all. Facilitated diffusion, again, it's still passive. It's allowing molecules to go through, but they require protein channels to do it. Okay? So facilitated diffusion is you can come through, but you have to use a channel to do it, as opposed to, say, oxygen that just easily diffuses right through the membrane. Now we'll talk about proteins that require energy, active transport to move molecules across. Okay, and there's many molecules that want to get from the inside of the cell to the outside, and some that want to get from the outside of the cell to the inside. So there's constant active transportation, and that's why cells need so much ATP for this constant movement. And all of this movement comes down to the same term, homeostasis, right? The ability of the body or the cells to maintain a stable environment, which is critical to survival. Now, when we're talking about water, water is going to move from a lower concentration to a higher concentration. So let's say the extracellular fluid has a high concentration of sodium. So Na. For whatever reason, your bloodstream and your extracellular compartment, which by the way are very much similar in terms of electrolytes, becomes very high in sodium. We call that hypernatremia. Well, so now what's going to happen is water from inside the cell, the cytoplasm, is going to want to go across the cell membrane via osmosis to try and dilute the extracellular compartment. And the cell is going to shrink in size. Okay? Let's do the opposite. What if you have low sodium outside of your cell? So you have a low concentration of sodium in the extracellular compartment, which typically means you have a low sodium in your intravascular compartment because they communicate and electrolytes go across blood vessels very easily. What's going to happen there is that the water in the extracellular compartment is going to go across the cell membrane and swell the cell, okay, cause cellular edema. And that'll come into play when we're talking about electrolytes and how they can affect whether the cell shrinks or whether the cell becomes swollen and edematous. Okay, so you can see how a low sodium in the extracellular compartment will cause water to, through osmosis, to go across the cell membrane and into the cell and cause cellular edema. Okay. So just see water as following concentrations of solutes. Okay, that's all it's going to do. And because water can easily go across the cell membrane, it's going to follow concentration gradients. Okay. Okay, so here's an example of facilitated diffusion. And it's showing ion channels and carrier proteins, right? So there's the extracellular space, so anything outside of the cells, And remember, when we're talking about the extracellular space, that's the distance between the blood vessels, say the little tiny capillaries that are bringing in oxygen and glucose and other nutrients to the area and taking away waste from the cells. Okay, that's the extracellular space. And then you have your plasma membrane or your cell membrane, and then the intracellular space. Okay, and when we're talking about diffusion, molecules are going to go down gradients. Some can go right across the plasma membrane, others use transport proteins. Okay, so some of them are channels, some of them are carrier proteins where they'll open on one side to let the molecule in and then open on the other side to release the molecule. Okay, so it's pretty amazing how the cell membrane can function this way, to have a variety of ways to allow different molecules to get in to the intracellular space, the cytoplasm. Okay, so you can also have facilitated diffusion with gated channels, right? So facilitated diffusion just means that there's transport proteins or transmembrane proteins that are allowing the molecules or the ions to come across the plasma membrane. They don't use energy. There's no ATP involved. So they can be ligand gated, meaning a molecule needs to attach to the protein and then open up the protein to allow the molecules to come across. Or voltage gated, meaning there needs to be some kind of an electrical current, a stimulus that then opens the transmembrane protein to allow molecules across. Or mechanically gated, meaning there needs to be some kind of movement of the channel or the transmembrane protein, which then allows ions across. So it's still not using any ATP. It's just different ways for membrane proteins to be controlled. So here's an example of a gated channel. So you have your protein, which is closed to sodium at the moment, and with some kind of an electrical change, for example, when the plasma membrane or the cell membrane depolarizes, the protein changes shape and allows sodium to pour into the cell. And this is partly how action potentials work, right? So sodium rushes into the cells to depolarize the cell, and potassium leaves the cell, and then the sodium potassium ATP pump is responsible for repolarizing the cell. Okay, so this is just one example. We spoke about mechanically gated or ligand gated, but they all have the same concept, that some kind of stimulus is required to then shift the protein into an open position, but no energy is required. Okay, and on this slide is just showing you passive transport. As soon as you see passive transport, it means there's no energy utilized for this process, right? So diffusion right across the membrane, no oxygen, no ATP required. And oxygen or CO2 would be an example. Facilitated diffusion, that would be, say, insulin required as a hormone to attach to a protein channel that will then allow glucose to come through the channel. So that could be a gated facilitated diffusion. But again, no energy used for facilitated diffusion. active transport, the main one we're going to talk about, but there's many others, is the sodium-potassium pump in the cell membrane. How the cell is able to actively pump sodium out and actively pump potassium into the cell using a transmembrane protein pump with ATP. Okay, so let's look at the sodium-potassium pump, which is in all cells of the body. And this is an example of active transport, right? So sodium is always higher in the extracellular fluid. It's more concentrated there. And it's at a lower concentration normally in the cell. The opposite is true for potassium. Potassium is usually at a higher concentration within the cell and a lower concentration in the extracellular fluid and the bloodstream. Okay, so when we see a serum potassium of 3.5 to 5, that's normal for the bloodstream and the extracellular compartment. That's not normal for within the cell. But when we talk about blood tests and what levels the sodium and potassium and chloride, we're really talking about the serum, which is the bloodstream. Okay, so this pump is responsible for pumping out sodium into the extracellular fluid and pumping potassium against a gradient, because potassium is at a higher concentration in the cytoplasm of the cell, pumping it into the cell. So this requires energy in the form of ATP. And ATP will be converted to ADP, and in that process it gives off some energy to run these pumps. Okay, so what's going to happen if, say for example, an area of your body that has millions of cells isn't getting any oxygen anymore? Say the blood vessel, the artery, is blocked to that area. So the area is now running low on oxygen. It's running low on glucose because the blood vessels, say the capillaries, are blocked. They're not able to deliver the nutrients to the cell. So the cell cannot process glucose because it's running low on glucose, and it can't produce ATP through aerobic metabolism. So the cell is going to have to convert to anaerobic metabolism, meaning the process called glycolysis that we'll talk about, and that just means the conversion of glucose to a molecule called pyruvate, but that doesn't make a lot of energy. So the body's going to do everything it can to make energy still, but without oxygen, it can't make a lot of energy. It's going to be anaerobic compared to aerobic metabolism with the oxygen. So when an artery or a blood vessel is blocked to an area, we call it ischemia, meaning lack of blood flow to an area, resulting in ischemia of the cell, reduced oxygen and nutrient delivery, and the cell is running out of energy, and the pumps will start to fail. When the pumps start to fail, potassium is now going to pour into the cell. Sodium is going to pour into the cell, and potassium is going to leak out of the cell. And when sodium pours into the cell, what's the fluid going to do? It's going to follow the sodium into the cell, and the cell is going to swell. It's going to cause cellular edema. And finally, as the cell gets bigger and bigger and bigger, it's going to burst. right? It's going to die. So that's how an infarction happens. So an infarction, such as a heart attack, which is a myocardial infarction, is cellular death. If we were talking about cells in the brain, we would call it a stroke, right? A cerebrovascular accident, a CVA. So when arteries get blocked, cells run out of nutrients such as oxygen and glucose. The pumps start not to work. Potassium leaks out of the cells, sodium leaks into the cells, the cells swell and become edematous, and then finally die. So the process of ischemia is that the cells are running out of energy and not able to metabolize and produce a lot of energy, but they haven't died yet. They're ischemic. They're in the process of potentially dying, but they haven't died. Infarction means that some cells have already died. Okay, so it's just a spectrum. Ischemia, and if the ischemia continues, then you get an infarction. Okay. Okay, so this is just showing you that there is an electrical potential across every cell membrane. Okay, so there's the resting state of about -70 millivolts, and that's because the sodium potassium pumps are actively pumping sodium out and potassium in, and they end up with a resting membrane potential. Okay, when a cell depolarizes, sodium rushes in, potassium goes out of the cell, you get this big spike of depolarization, and the membrane potential becomes positive temporarily. And then once the pump starts to pump the sodium out of the cell and potassium back into the cell, you get the repolarization phase. Okay, and it goes back to a resting state. Why do we care about this? Well, this is going to matter when we talk about, say, electrical conduction in the heart. Okay, how do nerves in the heart conduct electricity to then allow the heart to pump? Or electrical conduction along any nerves in the body. They do exactly this. They produce an action potential. Okay, and it runs down the axon of a neuron to the synapse, and if the synapse is connecting to another nerve, it'll communicate with that nerve, and another action potential will be created. Okay, so a resting membrane potential is really important for cells, and it's all about the sodium potassium pump. And remember, what happens if an artery dies that's feeding a large group of cells, say in the heart, so myocardial cells? If the artery gets blocked, those cells initially become ischemic, meaning they have a bit of time to survive. They're not dead yet, they're ischemic. And then if the blockage continues and no oxygen is being delivered to the area, they finally infarct. And that's why we call it, say, for example, a myocardial infarction. And that's the term for a heart attack. If we want to talk about another part of the body, say the brain, we could say a cerebral infarction, meaning a stroke. And the same thing has occurred. The cells have died because the pumps stopped working. Okay, so let's talk about the concepts of an agonist and an antagonist, because this is really important when it comes to drugs in pharmacology. Okay, so really have this concept well ingrained in your mind. So you have receptors in your cell membrane that are waiting for a signal to attach to them. And once, say, a molecule, or what we call a ligand, has attached to the receptor can then act in a certain way to have an effect within the cell itself. Okay, so the ligand, whether it's a hormone or some kind of molecule that's attaching to the receptor, and then it's allowing the receptor to become active and producing an effect within the cell itself. Okay, an agonist is typically a drug that behaves exactly like the endogenous ligand. The endogenous ligand is just like saying your natural molecule. So an agonist is a drug that's behaving exactly like the molecule that is made in your body to attach to that receptor. Okay? So it's going to cause basically the identical effect. What's an antagonist? So an antagonist is typically a drug that's going to go onto the receptor and block it. So the ligand can't attach to it. So even though another part of your body is trying to communicate and attach to the receptor and then have an effect within the cell, it can't happen because the receptor molecule has an antagonist on it. And we use agonists and antagonists all the time in pharmacology, okay? To have either the identical effect of a molecule that's typically in your bloodstream, or to have the opposite effect and block a receptor so that the endogenous molecule, the molecule that's made by your body, cannot attach at all. Okay? So let me give you an example. So say this is a receptor for a chemical in your bloodstream called epinephrine. So epinephrine is going to attach to a receptor. It's going to then have a stimulatory effect on the receptor, and the receptor is going to have communication within the cell to have a certain effect. Okay, so epinephrine is made by your body, and we can call it the endogenous ligand. Okay, what about an agonist? Well, an agonist is something that we can give a person to behave just like epinephrine. And in actual fact, we make epinephrine, so we could inject epinephrine into the body to have the identical agonistic effect. Or there's other drugs that we could use that behave very similarly to epinephrine, and they would be called agonists as well. Okay, so agonistic effects. What about an antagonist? Well, we can use a molecule or a drug that's going to block epinephrine's effects. And epinephrine cannot attach to that receptor any longer and exert its effect within the cell. Okay, so we could call it, say, an epinephrine blocker, or an epinephrine receptor blocker. Okay, so that's just the concept of agonist and antagonist, and this is definitely going to come up in pharmacology. So know the terms quite well. Okay, so let's just look at different ways that cells can communicate with each other, because it's very important for cells in one area to not only communicate with cells very close to them, but also with cells at a distance, right? So, for example, your brain wants to communicate with many parts of your body through nerves, okay? But also there's organs called endocrine organs that produce hormones that they excrete into or secrete into the bloodstream. And the pancreas would be a perfect example of an endocrine organ. It's an organ that secretes a hormone into the bloodstream. The pancreas actually also acts as an exocrine organ, meaning it also secretes digestive juices into the gastrointestinal tract. So external, so exocrine, internal, endocrine. So endocrine organs release hormones to go into the bloodstream, and there's many examples that we'll come to in other modules. Okay, so how does cells communicate with each other? Well, they can be attached directly to each other with gap junctions, which are just tunnels between cells, allowing molecules to pass between them. An autocrine communication is where a ligand or a molecule signals a communication within the same cell. A paracrine ligand is released from one cell that impacts a cell nearby. Okay. Endocrine we spoke about, those are organs that typically secrete hormones, which are molecules that go throughout your bloodstream and attach to other cells to communicate with them, what are called target cells. So you have a little gland in your brain called your pituitary gland. It releases a tremendous amount of hormones, and all those hormones go to different cells depending if the cell has a receptor for them. So they all go into the bloodstream, but the only cells that are going to respond are the ones that have receptors for the particular hormones. Okay? So when we say endocrine, we're really talking about hormonal, which is the next one, right? The hormone that's going into the bloodstream and going throughout the whole body and then attaching to receptors to have an effect. The neurotransmitters we spoke about at the beginning of this slide, they're just the nervous system, right? So a nerve will come to a synapse. It'll release a neurotransmitter such as acetylcholine or serotonin or dopamine. Those are very common neurotransmitters, and they'll cross the synapse to communicate either with another nerve that will continue the signal onwards, or the nerve will attach to a muscle and signal the muscle to contract. Okay, so it just depends on whether the nerve is attaching to another nerve or the nerve is actually going to a muscle. Okay, and this is just examples of the different chemical signaling, right? So the first one is self-signaling. So a molecule that signals the cell that it came from. You can have gap junctions where molecules cross from one cell to another. Paracrine communication, where a cell's signal will go to a very nearby cell. And endocrine, we spoke about a cell that secretes a hormone into the bloodstream, and it can have communication with cells all over the body at very distant places.